we discuss about Western blotting. It is also known as immunoblotting or protein blotting. It is laboratory technique used to detect and identify specific proteins from a complex mixture of proteins extracted from cells or tissues. Next principle, the principle of Western blotting involves separation of proteins by electrophoresis, their transfer onto a membrane and the specific detection of target proteins using antibodies and a visible or fluorescent signal. Next step, number 1, protein extraction and denaturation. Extract protein from the cells or tissues of interest using appropriate lysis buffer. Denature the protein by treating the sample with a detergent and heating it so that proteins unfold and gain a negative charge. Number 2, STS page gel preparation and loading. Prepare a polyacrylamide gel with different concentrations suitable for the range of molecular weight of the target proteins. Load the denatured protein samples into vials of the gel. Number 3, electrophoresis. Apply an electric current to the gel to separate the protein based on their size. Smaller proteins migrate faster and larger proteins move slow, creating bonds of separated proteins. Number 4, electroblotting, जिसे ट्रांसफ़र भी कहते हैं. Carefully transfer the separated proteins from the gel onto a solid spot membrane. This is achieved by applying an electric current which drives the protein out of the gel and onto the membrane while retaining their separation. Number 5, blocking. Block the membrane with a blocking agent to prevent non-specific binding of antibodies and reduce background noise. Number 6, antibody incubation. Incubate the membrane with a primary antibody that specifically recognizes the target protein of interest. The primary antibody binds to the target protein, forming an antigen-antibody complex. Number 7, washing. Wash the membrane to remove unbound primary antibodies, reducing background noise and non-specific binding. Number 8, secondary antibody incubation. Incubate the membrane with a secondary antibody conjugated to an enzyme of fluorescent tag. The secondary antibody binds to the primary antibody, amplifying the signal. Number 9, signal detection. Add a substrate for the enzyme conjugated secondary antibodies, or use appropriate equipment for fluorescent detection. The substrate or fluorescent tag generates a visible or fluorescent signal respectively, where the target protein is present. Number 10, data analysis. Capture the signal on X-ray films or use imaging equipment for fluorescent signal. Analyze and interpret the result to identify and quantify the presence of the target proteins. Next diagram. Next application, protein. It is used for protein detection and quantification. It is used to identify and quantify specific protein in complex biological sample. Diagnosis of disease. Clinically used in clinical setting for detection of certain disease markers and diagnostic purpose. Western blots can reveal protein interaction through immunoprecipitation experiments. Biomarker discovery. Western blotting aids in the identification of potential biomarkers for various diseases and conditions. Gene expression analysis. It can be used to study changes in protein expression in response to different treatment or conditions. Advantages इसकी क्या है? High specificity, sensitivity, versatility, and qualitative and quantitative analysis. Number one, high specificity. It allows the specific detection of individual proteins. Number two, sensitivity. It can detect low amount of target proteins in a sample. Versatility. It can be used with a wide range of antibodies and sample types. Qualitative and quantitative analysis. It enables both qualitative and quantitative analysis. Next, disadvantages. Time consuming. The process involves multiple steps and can be time consuming. Technical expertise. Western blotting requires skills and experience to obtain accurate results. Subjective interpretations. Band intensity interpretation can be subjective and may vary between users. And last, semi-quantitative. While it can provide relative quantification, it might not be as precise as some other techniques. Thank you so much.